Hi, and thank you so much for inviting me to speak at this event today. I'm going to be talking about brain waste disposal systems in dementia, or as I like to call it, brains, bins and bubbles. So you're all probably aware that across the different neurodegenerative diseases, you get this abnormal accumulation of protein, and this is variable across the different disorders. So for example, we've got tau, which is kind of a serial offender. It pops up in multiple diseases, including Alzheimer's and Parkinson's disease. But then some of the abnormal protein accumulation is more specific. So we've got FUS, TDP43, alpha-synuclein. But this abnormal protein essentially does not accumulate in a younger, healthier brain. And so that raises the question of why is this happening? And we know that this abnormal protein accumulation is damaging, right? It causes cell death, and it causes cell death across the spectrum of neurodegenerative disorders. So being able to interfere with that process of abnormal protein accumulation and to protect cells from death is something that I think across the board all dementia researchers are trying to achieve. Now, in a younger, healthy brain, we've got lots and lots of systems that are put in place to help stop abnormal proteins accumulate and cause damage. So just like at home, where we've got lots of different bins to help us have a nice, efficient and sustainable waste disposal mechanism, we've got a black bin for general waste, and we've got green bin, blue bin, brown bin, I think is what I've currently got outside my house. Our brains have got lots of different systems that serve different purposes as well. So we often speak about autophagy. And when we speak about autophagy, or when you see the word autophagy, what's usually been referred to is the process of macroautophagy. So macroautophagy is a non-selective bulk mechanism of waste disposal. It will just degrade without discrimination, worn out cellular components, damaged proteins, and it's quite a complex multi-step process as indicated in the image. And don't worry, you aren't supposed to be able to read all of that, but it's just to demonstrate really that it's a complex procedure with lots of different steps, lots of different cogs in that machinery that could potentially go wrong. Another type of autophagy is chaperone-mediated autophagy. So whilst macroautophagy is kind of more generic and it is involved in the non-discriminate disposal of waste, chaperone-mediated autophagy is more selective. So this is more like your brown bin, where you can put in, a bit of glass, bit of plastic, whereas your black bin, anything can go in there. So chaperone-mediated autophagy is dependent on its substrate proteins that are going to get degraded, having a specific pentapeptide sequence that is used to identify that specific protein for degradation. And there are other forms of autophagy as well. So microautophagy, for example, is more like your blue bin, where you can only put paper or paper-based products in. And there are lots of other types of waste disposal systems. So we've got the ubiquitin proteasome system. There's mitophagy, which is specifically associated with the degradation of defunct mitochondria. There's also astroglal-mediated interstitial bulk flow, which is very catchily named, isn't it? But all these systems are supposed to stop these proteins from building up in our brains. They're supposed to clear out any damaged components to stop them exerting any damage on the brain cell. So there's clearly something going wrong. Now, if we think about the spectrum of neurodegenerative disorders, over the last decade or so, there's been lots and lots of evidence that has been reported that shows that autophagy seems to be going wrong. The biggest body of research really focuses on macroautophagy, and I keep using that word autophagy, and that usually refers to macroautophagy. Whereas the other types of waste disposal system aren't as well researched. And macroautophagy and chaperone-mediated autophagy particularly work synergistically to clear abnormal proteins. Abnormal proteins such as those offenders that keep popping up in our neurodegenerative diseases. Now, if we think about, for example, frontotemporal dementia. Frontotemporal dementia is a really complex spectrum disorder where you've got heterogeneity, not just in how the disease manifests clinically, but how the disease manifests pathologically. So tau seems to pop up in a number of the different types of frontotemporal dementia, whereas FUS TDP43 is more specific and it has got a relationship to its underlying genetic component as well. But these disorders are complex, and you could predict then, because of the complexities associated with the overlapping pathology, the variation in the clinical syndromes, and also that background genetics, that you might have variation in the types of autophagy deficit that you might expect to see, what's actually driving or at least exacerbating that abnormal protein accumulation. Now, interestingly, when you look at the backgrounds, the genetic backgrounds of those disorders, many of the genes associated with frontotemporal dementia are linked either directly or indirectly to autophagy. So progranulin, C9orf72, CHUMP2B, these have been linked to either directly to autophagy and having a role in regulating and maintaining effective autophagy, or they've been linked to lysosomal function. We know lysosomes are very, very important to allow and facilitate the degradation of materials, and they're features of some of these autophagy and waste disposal systems. So we could predict then that, given that we've got a variation in how these disorders manifest, but we've got that commonality of we've got an abnormal protein building up and killing brain cells, we might predict then that there would be some variation in how autophagy might be disrupted. So this is something that has really driven the research in my group for years now. Why is this abnormal protein not being cleared? It should be being cleared. Hopefully, in my brain, it is being cleared. I like to think that anyway. And I showed you an image earlier which demonstrated the complexity of these systems, and purposefully not gone into it into too much detail given the time that we've got today. But these systems are complex processes, and there are lots of places, therefore, for things to go wrong. And that's kind of evidenced in these images here. So when you think about the process of waste disposal, and we think about what is going on in our brains, the damaged protein in the first instance needs to be identified as something that needs to be cleared. Otherwise, it's just going to litter the pavement and cause a whole heap of mess and damage. That damaged protein needs to be targeted and directed to the correct waste disposal system. If it isn't getting into the right waste disposal system, then that could mean that it doesn't get cleared, or it could mean that actually that system gets overwhelmed and the bin starts to overflow, or it might be that that waste disposal system just can't effectively clear the waste and break it down in the way that it's supposed to do. So there are lots of different places where waste disposal can go wrong in the brain, and we've got lots of different systems where waste disposal can go wrong in the brains. So what we decided to do was to have a look across a number of different types of dementia, so looking at Alzheimer's disease and looking at frontotemporal dementia, but broken down into its different genetic drivers. So frontotemporal dementia with granulin mutations, with C9orf72 mutations, or with tau mutations, to see if there was variation in some of the key autophagy markers across these different subtypes of dementia. And this work was funded by Alzheimer's Research UK, and it was lovely Richard Heal that did all this work. And what he did was he took brain tissue from the Manchester Brain Bank, and he looked at all different types of abnormal proteins that you might expect to see. From no diseased aging was kind of our control group across the spectrum of Alzheimer's and different types of frontotemporal dementia. So he looked at all the different abnormal proteins, such as He looked for tau, beta amyloid, P62, FUS, TDP43, and he also looked at these markers of autophagy as well. So LC3 is a common marker used to look at a macroautophagy. It's used to identify when you get the formation of an autophagosome, which is where all the action happens. Once the autophagosome has been formed, it then fuses with a lysosome, and then you get the degradation of the waste that's been internalized within the autophagosome. So LC3 is often used as a marker of macroautophagy, whereas LAMP2A is often used as a marker of chaperone-mediated autophagy. So LAMP2A is essentially a receptor on the lysosomal membrane, which is used to help attract the protein constituents that are going to get degraded. So these two markers are really common markers to look at macroautophagy and chaperone-mediated autophagy. I've just pulled out some examples of the data here because I think this tells a really interesting story. So this particular set of data is focusing on the frontal cortex, but we looked across different brain regions, some that were affected massively by neurodegenerative disease, others which are generally left alone, others which are impacted in some diseases but not others. But we're looking at the frontal cortex here because you do get involvement across the spectrum here in Alzheimer's and FTD. And what was interesting, you can see that from the bars here, the main thing that you can see initially is you've got variation. So you don't get an equal representation of those autophagy markers across our groups that we were interested in. And this is particularly evident in the white matter, actually. That's really interesting. So these autophagy markers, you see them in neurons, you see them in glial cells, and you can see specifically in some of the frontotemporal dementia groups, you can see a drop in those, particularly in LC3, a drop in those autophagy markers. And it was a different story, a very complex story, when we looked at the different brain regions of how these different autophagy markers were detected. And that kind of backed up our hypothesis that you get variation in how you would expect, variation in these autophagy markers at different parts of the brain which are differentially affected by disease, in the different groups which are differentially affected by different types of dementia. So it's a complex story. And that's really interesting and important when we're thinking about trying to develop therapeutics. If we just switch on autophagy, if we just turn it on, if we turn on macroautophagy, and we start the process of just chucking stuff into the bin, that could result in the bin overflowing and not actually having any therapeutic benefit. But if we can pinpoint exactly which system's going wrong, when, in which type of disease, then it means that we can be much more efficient and targeted with our therapeutic interventions. Now, running parallel to the study that Richard was doing, which was primarily focused on frontotemporal dementia, I had another student called Diana who was doing a study, and she was interested in Alzheimer's disease, and particularly in the progression of disease. So the work that she did also used tissue from the Manchester Brain Bank, and what she did was she looked at, she grouped cases for BRAC, so she had some low BRAC cases where you would expect to see kind of minimal tau and minimal accumulation of damage, shall we say, mid-BRAC group, and then later BRAC group. So we've got that spectrum from kind of no early disease to early disease to late disease. So the idea was to capture a spectrum of changes and avoid that kind of ceiling to floor effect of not much going on to look at here, a whole heap of pathology and weird stuff going on in the brain which is causing damage. So Diana was looking, she did the similar studies to Richard where she looked at the different markers across the different brain regions, across the different stages of disease, and found variation similar to Richard. But she also was interested very much on what was going on at a cellular level. And so Diana did some co-localization studies where she looked at some of these autophagy markers to see whether you could detect them within the cells that contained these abnormal protein accumulations, specifically tau we were interested in for this study. And what she found was as you went through the Braak stages, as with increasing Braak stage, you got a decreased co-localization between these proteins. So as you looked on a cellular level, sometimes there was a masking where you would see a similar amount of overall markers accumulating in some brain areas. So you say similar levels of LC3 or LAMP2A in the hippocampus between low and high Braak. But when you looked at a cellular level, it was just in a different place. So there was decreased co-localization. So in the cells where you got lots of abnormal tau, you didn't have as much of these autophagy markers. And I've always found that really fascinating. When you look at brain tissue, you'll see these brain cells that are jam-packed full of abnormal pathology, and the cell's sad and it's dying and it's not in a good place. And then right next to it, you will have a brain cell that it's not got any pathology in it, it's not got any abnormal accumulation. And I've always been interested in those microarray studies which see what's going on between the cells that are jam-packed full of pathology and the ones that seemingly healthy. Now the plot thickens here as well. So another thing that I've always been interested in since my PhD days was this evidence about the transsynaptic spread of disease pathogenesis. So when I was doing my PhD, I did some work which was mapping how tau spread specifically through the hippocampus, because we know so much about hippocampal circuitry, and how related it was to how the brain is connected and the pattern of deposition. If you look on a population level from no disease, early, mid, late disease, you get this spread of pathogenesis all the way through, so that it's linked to anatomical circuitry. That's really interesting. And at the same time, there were all these studies coming out from, for example, Matt Diamond's group, shown in the image here, where there was this evidence that extracellular tau could be taken up and it could seed and spread through synaptically to connected cells. Really interesting and exciting work, and it kind of led the way for lots and lots of studies to evidence this cell-to-cell spread of pathogenesis. But how's this happening? What's actually going on here? And that's where exosomes, or small extracellular vesicles as we now call them, could potentially hold the key in this. So small EVs are really, really interesting. They're released by all cells, neurons, glial cells, and small EVs hold all sorts of interesting things within them, different cargoes such as proteins, microRNAs, DNA, and we already know from other researchers that within small extracellular vesicles, some of our, shall we say, candidate pathogenic proteins such as amyloid fragments and tau have been already identified within EVs. And EVs can also cross the blood-brain barrier. So EVs could potentially tell us a lot about what is going on and what is driving disease, and therefore how we might be able to treat disease. And because they have the ability to be released from neurons and be taken up by neurons, they might underlie this transsynaptic spread of pathogenesis. So I've paired up with an outstanding genomic researcher called Dr. Arijit Mudapaka, who works at the University of Salford, and he for years has been studying small EVs in all different contexts, in brain tissue, in lung tissue. And so at the University of Salford there is this expertise in studying small EVs. So essentially from various bodily fluids, from cell culture, we've got the ability to harvest and separate out small EVs using size exclusion chromatography. We can also visualize EVs using electron microscopy and nanoparticle tracking analysis. We can test that we've definitely got EVs as opposed to other membrane-bound structures using Western blotting for specific EV markers, and also using fluorescent NTA. And we can also do some size profiling analysis, because we know the size of EVs is very specific, and we can use that to make sure that what we are looking at are definitely EVs. There's been some really nice work going on in our group over the last couple of years, really looking at the different neurodegenerative environments that are associated with neurodegenerative disease, such as oxidative stress, pro-inflammatory environments. And we've been looking to see how that influences the cargo of EVs, knowing that EVs contain microRNAs, DNAs, proteins. And we're specifically interested in microRNAs in our group. There's not been as much work gone on to microRNAs and microRNAs, lots of focus on the proteins. And so we're kind of moving back and to look at microRNAs, and that's where our interest lies currently. So what we've been doing in our group is looking at a number of different cell culture models. The model that I'm showing here is we use differentiated SHSY5Y cells, and we can grow these, we can extract the EVs, we can validate that we've got the EVs using the imaging, using the NTA and the Western blotting, and then we can use sequencing, next generation sequencing, to look at the microRNA content. And what we found so far is some really interesting findings about lots of different microRNAs that we're specifically interested, that are linked to some of the pathways that we're interested in, such as autophagy. We found that the different neurodegenerative driving environments, such as, for example, exposing cells to high levels of hydrogen peroxide, or not high levels, but levels associated with physiological oxidative damage, you get a massive change in the expression of some of our candidate microRNAs. Now, that's really, really interesting. So going forward then, what are we trying to do? Well, we're trying to connect the dots now. So thanks to some funding from the Alzheimer Society, the next phase of our work is going to be trying to put all this information together. So we've got lots and lots of data now from human brain tissue about the abnormal proteins that accumulate, and also the markers of the different autophagy systems. We've got some frozen brain tissue, and we've teamed up with Prof. Andrew Hill over in Australia, who has developed some expertise in extracting EVs from brain tissue, as well as the cell culture-based models that we're doing. And what we want to do now is to try to explore how autophagy is related to EV cargoes, and how autophagy influencing environments or influence EV cargoes. And this is the really juicy bit. If we look at different types of dementia, if we do a comparison between normal, non-diseased aging, shall we say, and Alzheimer's, and the different subtypes of frontotemporal dementia, is there a variation in those EV cargoes? And we've managed to bring on board some outstanding students. So we've got Joe and we've got Shannon, who have joined this project and are doing amazing work, even though they've only been here a couple of months. Why is this important though? So I've mentioned these EVs contain information. They contain some of our key pathogenic proteins. They've already been detected. And we also know that they contain the microRNAs. And all this could shed light on the abnormal accumulation of protein. And we know that the early diagnosis of dementia is really, really challenging. And this means it's difficult to get people in the right arm of the clinical trials to know what kind of medications are going to be useful in inhibiting that protein accumulation or clearing that protein accumulation. If we can shed light also on what is going wrong in the autophagy pathways, then again that gives us clues of what we could target therapeutically. So that is where we are going in the future. So I would like to thank all the wonderful people on this slide who have taken part in the research or are just starting on their research journey and have helped inform the direction of the work in the group. And also I'd love to thank the Alzheimer Society for funding our next adventure, and Alzheimer's Research UK, who funded the work previous to what we're doing now. And of course, Brains for Dementia Research. We use human brain tissue in the majority of our projects, which is supplemented with cell-based studies. And the University of Salford, who've also funded some of our work. And finally, I'd like to thank the Manchester Brain Bank, all the staff there are absolutely amazing and have helped shape some of the work that we've done, helped shape the analysis, and of course the donors of the families, because without that contribution, none of this would be possible. Thank you so much for your time, and I'll look forward to having any questions from you. I'm on Twitter as well if you want to get in touch, at Gemma Lace. So please do come and speak to me about all things brains related. Thanks for your time.